Hello everyone. Today we are discussing this question from GATE examination 2018 paper Excel-Q biochemistry question number 12. And my name is Shurosri Pal. I am a Prime Minister's research fellow. The question 12 states match the protein elution condition given in group 1 with the appropriate chromatography matrices from group 2. In group 1 there is P increasing concentration of sodium chloride, Q increasing concentration of histidine, R decreasing concentration of ammonium sulfate, and S decreasing concentration of H plus. In group 2, there is number 1 phenylsaccharose, number 2 chromatofocusing, number 3 DAAE cefacryl, and number 4 nickel NDA. And the question is to match group 1 with group 2. So we would like to discuss a bit about these chromatographic techniques. First of all, we will talk about this nickel NTA chromatography. So nickel NTA chromatography is an immobilized metal ion affinity chromatography, or we can also call it IMAC. So here on a support, this kind of a support can be saccharose or agarose. On it, this nickel, um, um, this actually nickel ion is immobilized, and this nickel ion is immobilized on this NTA support. So the NTA is nitrile triacetic acid. So we can see there are three acetic acid moieties here. These are the three acetic acid moieties, and on these three nitrile triacetic acids, uh, this is actually joined with a nickel atom. So we know nickel atom can form coordinate bonds, and this nickel atom can form six coordinate bonds. Among them, we can see that this nickel is um, joined with four coordinate bonds. So there are four coordinate bonds here, which joins the nickel atom with the NPS support, and two of its coordinate bonds are free. These two coordinate bonds can form with the imidazole ring on the protein structure. Now, where is this imidazole ring present in the protein structure? So, if you look at the structure of the amino acid histidine, we know that in the side chain of the amino acid histidine, we can find a imidazole ring. And this nickel atom can actually form coordinate bonds with this imidazole ring structure, which is present in the amino acid histidine. So, for using this kind of a chromatographic setup, where we are basically depending on the affinity of this nickel atom with the uh, imidazole ring. So that is why it is a type of affinity chromatography. And for using this type of affinity chromatography, we tag our proteins with 6x polyhistidine tag. So what is 6x polyhistidine tag? It basically codes for 6 histidine amino acids. So these tags are usually in the vector where we clone the uh, gene of our protein of interest, right? And so this vector will express the histidine tag either on the N-terminal end of the protein or on the C-terminal end of the protein. And because of that, this protein will have this 6x histidine tag. So it will have consecutive six um, histidine amino acids either on its N-terminal or on its C-terminal. And because of that, these protein can bind to this nickel NTA matrix, and depending on that affinity, we can purify the protein. So, for example, this is the matrix, and we have this protein sample. So, suppose this is cell lysate. We have lysed a cell, and we have isolated all the proteins out, and in this cell there will be different types of proteins, um, but only our protein of interest will have this polyhistidine tag on it. So this is our protein of interest, so which has this histidine tag on it. And when we pass this protein sample through this column, through this matrix, the histidine tagged proteins will bind to this nickel matrix, and this affinity can be actually reversed by um, passing imidazole, increasing concentrations of imidazole. So by by passing increasing concentration of imidazole, we can actually elute this bound protein from the column, and that is how we can purify the protein, right? So these proteins are now purified, which has this histidine tag. So for elution in this kind of nickel NTA chromatography, we use imidazole, increasing concentration of imidazole. So why do we use imidazole? Because it has this imidazole ring which can bind to the nickel NTA matrix, and this imidazole can actually replace our protein from the column, so that the protein which has previously bound to the nickel NTA matrix can now be eluted out. So if we go back to the question, nickel nickel NTA chromatography here can match with the elution condition, increasing concentration of histidine. So histidine or imidazole can be used for elution of protein from nickel NT matrix. So Q will match with 4. Q will match with 4. Now the next type of chromatography that we are going to discuss is phenylcephalose. Phenylcephalose. So phenylcephalose is actually a hydrophobic interaction chromatography matrix. What is hydrophobic interaction chromatography? It is a type of chromatography that separates proteins according to differences in their surface hydrophobicity. So we know that in the proteins there are hydrophobic amino acids and there are hydrophilic amino acids. So the hydrophobicity on the surface of the protein, that is used as a parameter for separation in the hydrophobic interaction chromatography. And it utilizes a reversible interaction between the proteins and the hydrophobic ligand of a HIC resin. So the matrix it can be saccharose or it can be agarose. This matrix uh, is coated with ligands which are hydrophobic, right? So these are some of the HIC ligands. So these can range from hydrophilic to hydrophobic, and these ones, close, if you go down, the hydrophobicity actually increases. So starting from ether, PPG, phenyl, butyl, hexyl. So hexyl is the most hydrophobic one here. Right, and the one that we're talking about is phenyl saccharose. So basically, when saccharose is coated with phenyl groups, so the interaction between the hydrophobic proteins and the hydrophobic chromatography resin is greatly influenced by the running buffer. So the buffer, if it has high salt concentration, it enhances the interaction between the um, hydrophobic matrix and the hydrophobic proteins. But if you lower the salt concentration, that will, if you lower the salt concentration, that will weaken the interaction. So by lowering the salt concentration, the hydrophobic proteins which are bound to the matrix can be diluted out of the column. So proteins with different degrees of surface hydrophobicity can be separated using hydrophobic interaction chromatography. The proteins are bound to the hydrophobic ligand on the hydrophobic chromatography resin in a binding buffer with high salt concentration. So, the initial buffer, the starting buffer, usually has high salt concentration. So, that is how we equilibrate the column also. So, we prepare the column with high salt starting buffer. Then what we do, that we pass the protein sample through it. So the target molecule binds to the ligand on the resin via hydrophobic interactions. The sample buffer needs to have the same pH and ionic strength as the starting buffer. So we know the protein sample should be in the starting buffer. And now coming to the elution, so when the ionic strength of the buffer is reduced, the interaction is reversed. So the protein with the lowest degree of hydrophobicity will be eluted first. The most hydrophobic protein will elute last, which will require a greater reduction in the salt concentration. So here there is one example of four proteins: cytochrome C, RNase A, lysozyme, and alpha chymotrypsin. So here yellow colors indicate the hydrophobic residues. And if you can notice, cytochrome C has least surface hydrophobicity, whereas alpha chymotrypsin has highest surface hydrophobicity. So, which is why when we pass this protein mixture through hydrophobic chromatography resin, we see that when we actually reduce the salt concentration, the most hydrophobic protein will elute last. So, which is why alpha chymotrypsin is eluted last, right? And this is the salt concentration. This orange line is for the salt concentration. So, we can see that the salt concentration is being reduced, and the least hydrophobic protein will be eluted first. So, that is what happens with cytochrome C, followed by RNase A, followed by lysozyme, and followed by the most hydrophobic protein here, which is the alpha chymotrypsin. So, elution should be done with decreasing salt content, and uh, then that will cause the hydrophobic proteins to elute. And regeneration, so remove all the molecules to ensure full capacity that the resins are available for the next run. We should clean the resin with zero point 0.5 to 1 normal sodium hydroxide. So this is hydrophobic interaction chromatography, and the matrix that can be used for such a chromatography is phenylcephalose, and the elution conditions should be decreasing concentration of salt. So going back to our question, phenylcephalose will therefore match with decreasing concentration of ammonium sulfate. So we know that ammonium sulfate is a salt, and if we decrease the concentration of ammonium sulfate, the hydrophobic proteins will be eluted from the hydrophobic interaction chromatography resin. So R will match with 1. Now we will look at chromatofocusing. What is chromatofocusing? So chromatofocusing is a separation technique that allows separation of a single protein from a complex mixture based on differences in their isoelectric point. Chromatofocusing is similar to, we can say it is similar to ion exchange resin, and it is also typically performed on FPLC system. FPLC is fast protein liquid chromatography system. Um, In chromatofocusing, what happens? The bound molecules are eluted by altering the pH of the buffer, which results in the net surface charge of the bound molecules. And as the change in the pH of the buffer system crosses the pi of a given molecule, so when the pH is greater than pi, this molecule will elute from the resin as it has no net surface charge. So simply, this technique focuses on the isoelectric point of the proteins. And this technique, this chromatofocusing, can be used as a polishing step. Um, so when some to purify the protein from a mixture of proteins. And so these it can be from a very similar protein species which differ only by zero point zero 0.02 pH units that may not separate well using traditional ion exchange method. So in ion exchange, we will talk about it later, that in ion exchange, we also separate the proteins according to their net surface charge. So in a particular pH of buffer, whether the protein is positively charged or negatively charged. So um, depending on that, we actually perform ion exchange chromatography, and whether it is a cation or anion, based on that we choose our matrix. Chromatofocusing is also very similar to ion exchange, but just the difference is that uh, it gives a better resolution than ion exchange, because it can separate proteins which differ only by zero point zero 0.02 pH units, and also um, it uses ion exchange chromatography, it depends on the isoelectric point of the proteins. So chromatofocusing combines the advantage of high capacity ion exchange procedure with the high resolution of isoelectric focusing with a single strip procedure. So this is a typical chromatofocusing technique. The sample was partially purified hemoglobins. The column was mono P, so it's a polystyrene column. And so the start buffer and the elution buffer, we have to carefully look at it. So in the start buffer, the pH is 8.1, and this is the flow rate 1 ml per minute. And in the elution buffer, we use the pH 6.65, right? So what we do is we uh, decrease the pH of the buffer. And so if we decrease the pH of the buffer, that means we increase the um, H plus ion concentration, right? So by decreasing the pH of the buffer, so we can see it here that we start with pH 8. From that, we decrease the pH to pH 6.5. And in that, the proteins are eluted, right? So these are the four proteins that are eluted. And so that this actually depends on the pi of the protein, or the isoelectric point. So if we go back to our question again, chromatofocusing will therefore we can match it with decreasing concentration of H plus or increasing the pH. So S will match with 2. And the last one, which is left, that is the DEAE separated. What is DEAE? DEAE stands for diethyl aminoethyl, and this is a ion exchange resin. So we know what is an ion exchange chromatography. In an ion exchange chromatography, the matrix is either positively charged or negatively charged, and the proteins uh, that we want to separate, that will also have a particular charge, either positive or negative. That will depend on the pi of the protein and also on the pH of the buffer. So there is a mixture of proteins. For example, this column, we can see that the matrix is positively charged. So when the matrix is positively charged, anions will bind to it, right? So therefore, this matrix is called an anion exchanger. So DEAE is an anion exchanger. So this is the structure of DEAE, diethyl aminoethyl. So this is attached onto a cellulose support. So this can be a DAE cellulose, or if it is cefacryl, then it is called DAE cefacryl, like that. So now this is an anion exchanger. So it means that the matrix is positively charged and it can exchange anions. So like this one, this matrix is positively charged, and therefore the negatively charged proteins will bind to this matrix. And the positively charged proteins will not bind to the matrix. So they will come out uh, in the flow through, right? So now what will happen after the flow through is collected, then we will add the elution buffer. So basically what we do here in ion exchange, we increase the concentration of the salt in the buffer to elute the protein from the ion exchange matrix. So addition of elution buffer with increased concentration of salt to separate the highly negatively charged molecules. So when we increase the salt concentration in the elution buffer, the electrostatic interaction between the matrix and the protein bound proteins will be disrupted, and the protein will be eluted out from the column, right? So basically, it is a technique for separating compounds based on their net charge. Ion exchange chromatography media contain negatively or positively charged functional groups covalently bound to a solid support, either yielding a cation exchanger or an anion exchanger. So in a anion exchanger, the matrix is positively charged. In a cation exchanger, the matrix is negatively charged. And the charged compounds are absorbed or retained by an ion exchanger having the opposite charge, whereas compounds that are neutral or have the same charge pass through the void volume and are eluted from the column. So, like these ones, which are which have similar charge with the matrix, they are eluted out in the void volume. And the binding of the charged compounds is reversible, and adsorbed compounds are commonly eluted with a salt or pH gradient. So we increase the salt concentration, or we increase the pH or decrease the pH, and then the proteins can be eluted out. Um, so this is ion exchange chromatography, and so this DEAE separately will actually match with increasing concentration of sodium chloride by increasing the salt concentration. All right, so here P will match with 3, P matches with 3, Q matches with 4, Q matches with 4, R matches with 1, and S matches with 2, right? So therefore, our correct answer is option A. All right, so that's it, and thank you.